Hi everyone, welcome back to my YouTube channel and today we are going to talk about prokaryotes. So let's start our topic with introduction. Prokaryotes represents the earliest and most primitive form of life on earth. They are unicellular organisms that lack a membrane-bounded nucleus and other organelles. Despite their simple structure, prokaryotes are remarkably diverse, highly adaptable and essential to main biological processes and ecosystem. Study of prokaryotes form the foundation of microbiology and help us understand basic life processes, evolution, and biotechnology. Definition of prokaryotes. Prokaryotes are microscopic unicellular organisms that lack a true nucleus, and they are membrane-bounded organelles. They form the term prokaryotes from the Greek word that is pro, that means before, and karyon, that means nucleus, indicating that these organisms evolved before the development of defined nucleus. Their genetic material is found in a single circular DNA molecule located in the nucleoid region of the cell. Major groups of prokaryotes. Prokaryotes are classified into two major domains. First is bacteria, that is the most common and widely distributed group. It includes both beneficial and pathogenic bacterial species. It is found in soil, water, air, and inside living organisms. Second group of prokaryotes are archaeans that are structurally similar to bacteria but genetically and biochemically very different from them. They often live in extreme environments such as hot springs, salt lakes, and acidic environments. Characteristics of prokaryotic cells. Prokaryotic cells possess several distinguishing features, including they have no nucleus, as DNA is located in the nucleoid region. Second, they do not have membrane-bounded organelles, as the functions of them are carried out in the cytoplasm. Third, they have smaller size, that is typically 1 to 10 micrometers in diameter. Fourth, they have cell wall that is present in most prokaryotes, providing structural support and protection. Fifth is plasma membrane, as they control the entry and exit of substances. Sixth, they have ribosome that is present for protein synthesis, though they are smaller than the ribosome that are present in eukaryotes. Seven, they have binary fission, that is a simple process of cell division for reproduction of prokaryotic organisms. Now let's talk about structure of prokaryotic cells. A typical prokaryotic cells include the following component. First is cell wall that provides shape and structural integrity. In bacteria, it contains peptidoglycan. In archaea, the composition of the cell wall is different. Second structural component is plasma membrane, that is semi-permeable membrane that surrounds the cytoplasm. It controls the transport of nutrients, waste, and gases. Third component is cytoplasm, that is a jelly-like substance where metabolic reactions occur. They contain enzymes, nutrients, and ribosomes. Fourth structural component is nucleoid, that is a region where the cell circular DNA molecule is located. It is not enclosed by membrane, like in eukaryotic cells. Next structural feature is ribosomes, that are site of protein synthesis, that are smaller and simpler than eukaryotic ribosomes. Next structural component is flagella, as you can see here in the diagram. It is long whip-like structure that is used for movement in some species of prokaryotes. Next, they also have pili or fimbriae, as you can see here in diagram. They are hair-like projection that help in attachment to surfaces and conjugation, that is basically DNA transfer mechanism for the diversity. Next structural component is capsule, that may be present or may not be present. It is a protective outer layer that is found in some bacteria. It helps in adherence to surface and evasion of host immune responses. Reproduction in prokaryotes. Prokaryotes reproduce asexually through binary fission. They have their circular DNA replicate first of all, then their cell grows and DNA copies move to the opposite end of the cell. Then the cell membrane pinches inward and divides into two identical daughter cells. Some prokaryotes also exchange genetic material through processes such as conjugation, that is, DNA transfer through a pilus. Second, there may be genetic material exchange by transformation, that is, uptake of DNA from the environment. Third, there may be transduction, there is DNA transfer through bacteriophages. These mechanisms contribute to the genetic diversity between the prokaryotes and as they lack the property of sexual reproduction. So this was all about reproduction in prokaryotes. Now let's talk about metabolism and nutrition of prokaryotes. Prokaryotes have a wide range of metabolic strategies. First, they may be autotroph that produce their own food like photosynthetic bacteria. They may be heterotroph that obtain nutrients by consuming organic material, and they may be aerobes that require oxygen for respiration, or anaerobes that live without oxygen and may be harmed by its presence. Some of the prokaryotes or chemotrophs that obtain energy by oxidizing inorganic substances such as ammonia or sulfur compound. Ecological and biological importance of prokaryotes. Prokaryotes play essential role in biosphere. First is nutrient cycling. As they decompose organic material, recycling nutrients like nitrogen, oxygen, and sulfur. Second importance is symbiosis, as they live in mutualistic relationship with plants, animals, and humans, like gut microbiota is an example of symbiosis. Third significance is biotechnology, as they are used in genetic engineering, fermentation, bioremediation, and antibiotic production. Fourth, they are used in disease, as some bacteria are pathogenic and cause disease such as tuberculosis, pneumonia, and cholera. And despite their harmful effect of some, many prokaryotes are harmless or beneficial. Prokaryotes versus eukaryotes. Prokaryotes differ from eukaryotes in the following major aspects. As prokaryotes have no nucleus organelles, they have DNA that is circular and located in nuclear region. Third, their cell division occur by binary fission, not by mitosis. Fourth, their ribosomes are smaller than the and structurally different from that of the eukaryotic organism. Fifth, prokaryotic cells are generally smaller and simpler, and understanding these differences is crucial in microbiology, medicine, and biotechnology. So you have to learn about these differences. Next, we will talk about evolutionary perspective of prokaryotes. Prokaryotes are believed to be the first form of life on Earth that appeared over 3.5 billion years ago. Their simplicity, resilience, and adaptability allowed them to thrive in diverse and extreme environments. Some prokaryotes, especially cyanobacteria, are thought to have played a key role in oxygenating Earth's atmosphere through photosynthesis. Conclusion of this lecture video. Prokaryotes are simple yet powerful organisms that have shaped life on Earth for billions of years. Their ability to adapt, reproduce quickly, and perform a vast array of metabolic functions make them vital to ecosystem, human health, and industrial application. While often overshadowed by more complex eukaryotes, prokaryotes remain a fundamental to understanding the life and advancement of science and technology. And this was all about prokaryotes. Hopefully you liked the video. If you do, don't forget to like, share, subscribe, and comment. Thank you for watching.